Greetings and welcome to our video on meiosis, Punnett squares, and genetic variation, explained by Dr. Worth and McChickenMan. We will visualize the process of meiosis, chromosomes crossing over, Punnett squares, complete dominance versus incomplete dominance versus codominance, and blood type inheritance. We begin our story with a sex cell at interphase. It grows and DNA is replicated. But the journey of meiosis truly begins at prophase 1. Chromosomes coil and thicken and become distinct. The nucleolus dissolves and centrioles move to opposite poles, forming spindle fibers as they go. The chromosomes form homologous pairs, which means each chromosome in the pair have the same genes, but different expressions of those genes. One homologous chromosome came from the mother, and the other from the father. During prophase 1, these pairs cross over, creating genetic variation. The next phase is metaphase, in which the spindle fibers push and pull the homologous pairs of chromosomes to the center of the cell. Anaphase 1 comes next, during which the homologous pairs of chromosomes are separated, and the spindle fibers pull the chromosomes to opposite poles. During telophase 1, the nuclear membrane returns for a short time, and cytokinesis occurs, splitting the cytoplasm and forming two unique cells. The next set of phases are unique to meiosis. The first phase is called interkinesis. The cells grow, but DNA is not replicated. Both cells undergo prophase 2, which is similar to prophase 1, but no crossing over occurs. In metaphase 2, the chromosomes line up. In anaphase 2, the sister chromatids are separated, and in telophase 2 and cytokinesis 2, the two cells split into four unique cells called gametes. The forming of the gametes marks the end of our journey. Gametes are the two required components of sexual reproduction. The gametes are unique because of the crossing over during prophase 1. And now you know, truly, how babies are made, and why everyone is unique. Now we are going to demonstrate the crossing over of homologous chromosomes during prophase 1. The homologous chromosomes line up and form tetrads, which consist of four chromatids. The chromatids within a tetrad twist around each other, with portions breaking off and attaching to adjacent chromatids. The result is variation of genetic information, because each chromosome has a different set of information that is distributed among four gametes when meiosis is complete.
Punnett squares were invented by R.C. Punnett in 1906. Punnett squares visualize all the possible genotypes of offspring. This is a Punnett square. The alleles of the first parent go here, and the alleles of the second parent go here. Today, we will be studying the possible genotypes of blubs. Green color is dominant to yellow color. Both blubs are heterozygous, having alleles for both traits. We'll put representations of the alleles besides the letters we are going to use for them. We'll use G to represent the color trait of blubs. In the first box, we see that both alleles are for the green color, so the offspring would be homozygous for the green trait and be green. And in the second and third boxes, we see that the green trait overpowers the yellow trait, because yellow is recessive. The offspring would be green and heterozygous. In the fourth box, both alleles are recessive, so the offspring would be homozygous for the yellow trait and be yellow. In the last example, we demonstrated an example of complete dominance. Now we will show other kinds of dominance. We now have a species called blebs. The color of blebs is determined by incomplete dominance, which means neither allele is completely dominant nor completely recessive. Here we have two blebs. Both are heterozygous for the possible bleb colors. We'll use Z1 to represent the yellow trait, and Z2 to represent the blue trait. In the first square, we have two yellow alleles, which means the offspring would be yellow. In the fourth box, we have two blue alleles, which means the offspring would be blue. But in squares 2 and 3, this is where incomplete dominance differs from complete dominance. Because neither allele is fully expressed, they show up as a mix between the two, making the offspring of these blebs green. The last type of dominance we will cover is codominance, which means both traits are expressed equally. This time we will study blorbs. The color of blorbs is determined by codominance. Here we have two heterozygous blorbs. We will use T1 to represent the black trait and T2 to represent the white trait. Like all the other squares, two of the same alleles means you get homozygous offspring for the same trait. The main difference of codominance is that both traits are fully expressed, meaning the homozygous offspring of blorbs are marbled black and white.
Warning: the following content contains blood. Viewer discretion is advised. Blood type is an inherited trait, which means we can plug it into a Punnett square. Antigens determine blood type, which determines whether or not you can accept a blood transfusion, as your body makes antibodies for the antigens your body does not have. Here we have a Punnett square again. Punnett squares are your friends. You must embrace them. You cannot escape them. Martin and Charlotte are a happily married couple. They want to know the possibilities of the blood types of their children. Martin's blood type is A. His genotype is AO. Charlotte's blood type is B, and her genotype is BO. We will put Martin on the top and Charlotte on the side. In the first square, we would have the blood type AB, which means the child would have both antigens and can accept any blood type. In the second square, the genotype is BO, which means the child would have B antigens, A antibodies, and B-type blood. In the third square, we have AO, which means the child would have A antigens, B antibodies, and have A-type blood. The last square has OO, which means the child would have A and B antibodies and have O-type blood. They would be able to donate blood to anyone. Thank you to Mrs. Riley for teaching our biology class. Thank you for watching our biology presentation. If you liked the video, press the like button. Subscribe. If not, well, prepare to be a donor yourself. Punnett squares are your friends.